Hi, welcome for continuation of this series. So, vacuole, as we can see here, normally is found in both plant and animal cells, but in plant cells, a vacuole is very large. It's very large and it stores material. So, a vacuole, a vacuole is a fluid filled. It is a fluid-filled sac, as we can call it, uh, used for the storage of materials needed by the cell. Used for the storage of materials needed by the cell. So we can call it a store. It acts like a storage or a godown. So these materials may include water. Materials here may include water, example, water. So they may store water. Uh, they may also store food molecules. They may store food molecules, inorganic ions, inorganic ions, and enzymes. So if they store all these, then they are very essential or very important to the organisms. So most animal cells contain many small vacuoles. So animal cells, animal cells contain many. So animal cells will contain many small, uh, many small vacuoles. But the plant cells normally have, uh, in plant cell, we have the central vacuole, which is a structure unique to plant cells. So it is a single large vacuole. So in plants, we have a single large vacuole. So this vacuole has its own advantages. It has its own advantages. It is a single large vacuole that usually takes up most of the space inside a plant cell. So it is filled with watery fluid. It's filled with watery fluid. It is filled with watery fluid. This fluid has its own advantage that strengthens the cell and helps to support the entire plant. That strengthens the cell and help to hold and support the entire plant. Help to support the entire plant. Helps to support the entire plant. So when a plant wilts, its leaves shrivel because there is not enough water in each cell's central vacuole to support the leaves' normal structure. So the central vacuole may also contain other substances, including toxins that would harm predators. So it may contain also toxins that would harm predators, because we know that plants don't have a way of avoiding predators. They can only do this by storing these toxins. Uh, Waste products that would also harm the cell itself would be released and stored here. Also, waste products. So waste products from the cell itself, and pigments that give color to cells, and pigments that give color to cells are also stored here, such as those in petals or flowers, such as those or in petals of flowers. The next organelle that we are going to look at is closely related to to the vacuoles, and this is just but the lysosomes. So the lysosomes, what they do normally, they will digest and recycle foreign materials or worn out parts. So these ones act like uh, the cleaners of the cell or the garbage collectors. So their main function is to clean the cell of any debris. So they contain enzymes. These contain enzymes. They contain enzymes. They contain enzymes. So you can guess that for them to contain enzymes, these enzymes have a purpose. And the main purpose is they defend the cell from invading bacteria and, and virus. They defend the cell from invading bacteria and viruses. So invading bacteria and viruses will be destroyed by the lysosome. So they also break down damaged or worn out cell parts. They break down damaged or worn out cell parts. So as I say, they act like cleaners. All the worn out parts, they will damage them. So lysosomes tend to be very numerous in animals. They tend to be very numerous in animals. Their presence in plants is still questioned by some scientists, but others assert that plant cells do have lysosomes, though fewer than are found in in animal cells. So recall that enzymes are proteins. So initially, lysosomal enzymes are made in the ER, in an active form. Lysosomal, so lysosomal enzymes are made in the ER. They are made in the ER, that is the rough endoplasmic reticulum, in an active form. In an active form. Vesicles pinch off from the ER membrane, carry the enzymes, and then fuse with the Golgi apparatus. There, the enzymes are activated and packaged as lysosomes. So these enzymes are activated, uh, the enzymes from the ER are activated and packaged into vesicles in the ER. I'm not in the ER, but in the Golgi apparatus. In the Golgi membrane. In the Golgi apparatus. There, the enzymes are activated and packaged as lysosomes that pitch off from the Golgi membrane. I mean the Golgi membrane. So they pitch off from the Golgi membrane in an active form. They pinch off from the Golgi membrane. This time in an active form. Active form. So the lysosomes can then engulf and digest target molecules. When a molecule is broken down, the product passes from the lysosomal membrane and into the cytoplasm, where they are used again. So what has been broken down by the lysosome is released into the cytoplasm. So lysosomes provide an example of an importance of membrane-bound structure in eukaryotic cell. So because lysosomal enzymes can destroy cell components, they must be surrounded by a membrane. Because lysosomal enzymes can destroy cell components they must be surrounded by a membrane. They must be surrounded by a membrane. I think it's just logical that this membrane normally will protect the cell from self-destruction. So this is just logical. So uh, surround this membrane that prevents from destroying necessary structures. However, the cell also uses other methods to protect itself from these destructive enzymes. For example, the enzymes do not work well in the cytoplasm as they do inside the cytoplasm. So these enzymes, even though they can digest cell components, they can't work well when they are in the cytoplasm, but rather they work best when they are inside the lysosome. So when they are inside the lysosome, they work best. But when they are released into the cytoplasm, they don't work good. So cell function depends mainly on also where where it works best. So these enzymes work best inside the lysosome and not in the cytoplasm. The next organelles that we will go through are the centrosome and centrioles. So as you can see from the picture, this is a top view. As you see from the top, you can see these are the centrioles here. And as you see side view, you can also see them. So centrioles divide DNA during cell division. So during cell division, they will normally be the ones which are responsible for dividing the DNA. So the centrosome is a small region of the cytoplasm that produces microtubules. It is the small, just a small region of the cytoplasm that produces microtubules. That produces microtubules. We saw the importance of microtubules in an earlier discussion. We saw that they aid or help in maintaining cell structure by maintaining a cellular skeleton. So in animal cells, it contains two small structures called centrioles. In animal cells, in animal cells, it contains two small structures called centrioles. So what is the function of the centrioles? So centrioles are cylinder-shaped organelles made of short microtubules arranged in a circle. The two centrioles are perpendicular to each other. They are perpendicular to each other, as you can see in the figure here. By perpendicular, we mean you can see them. They they seem to be in the opposite directions. So before an animal cell divides, the centrosome, including the centrioles, double, and the two new centrosomes move to the opposite ends. So before cell division, the centrosomes will double. Before cell division, before cell division, the centrosome doubles. So just like all the other uh, organelles that double, like the DNA and the others, it also doubles. So what happens as the centrosome duplicates, normally we will find that we will have the two centrosomes on different directions of the cell, and the spindle fibers, it will develop microtubules. These microtubules will grow and develop to what we call normally spindle fibers. the spindle fibers. Sorry, spindle fibers, which are important, of course, during cell division in pulling apart or dividing the DNA molecule. So centrioles were once thought to play a critical role in animal cell division. However, experiments have shown that animal cells can divide even if the centrioles are removed. Animal cells can divide even if the centrioles are removed. The centrioles are removed. So the belief that the centrioles must be there for an animal cell to divide has been displaced by, of course, research. So, uh, and normally animal cells, in addition, although centrioles are found in some algae, they are not found in plants. They are not found in plants. So centrioles are also organized microtubules to form cilia and flagella. Centrioles also organize. They organize microtubules. They organize microtubules to form cilia and flagella. To form cilia and flagella. To form cilia and flagella. So cilia look like hairs, flagella looks like a whip or a tail, and their motion forces liquids up past a cell. So for single cells, this movement results in swimming, but for cells anchored in tissue, like in our respiratory tract, the motion sweeps liquids across the cell surface and pushes substances out. So normally in our in our throat, we have these uh, cilia which sweep away particles which will have entered the respiratory tract, and as we cough, we can release them out. As we cough, we can release them out. The next important organelle that we are going to discuss about that is very important is the cell wall, but I would rather discuss it so that in another video, we, we will look at it. We will look at the plant cell. I mean the chloroplasts.